Bismillahirrahmanirrahim. Let's talk about virology. Note that this little topic will cover the main points to be known about virus. For example, the structure of virus, whether icosahedral or helical, positive stranded or negative stranded, DNA or RNA virus, single stranded or double stranded, enveloped or non-enveloped spectrum. DNA virus, positive stranded and icosahedral DNA virus. So naked and enveloped. For naked, mnemonic is APP. A P P P. APP. मतलब P is three times. Adenovirus, parvovirus, papillomavirus, polyomavirus. Right? Enveloped mnemonic her happy pox. Her herpes virus जिसमें HSV1 to herpes simplex virus, HPV, VZV like varicella zoster, Epstein-Barr virus, cytomegalovirus, and HHV 6 and 8. Second, happy से क्या hepatine virus, hepatitis B virus, pox virus, all pox viruses like smallpox, cowpox, not but not chicken pox, which is varicella zoster. That is a herpes virus. So okay, varicella zoster virus is also enveloped virus. Some important points about DNA virus family. In boards exam, most of time the family of virus is asked. If you know the DNA viruses, you can easily recognize which one is RNA virus. RNA viruses have got a big family and it is easy to memorize DNA virus and just to exclude which one is RNA or to recognize DNA virus among RNA viruses. All the DNA viruses are stranded, just like RNA. Just like DNA is double stranded, except parvo. This is important. So remember, all DNA viruses are double stranded except parvovirus, which is single stranded. And remember, if one strand is removed from it, it makes the parvovirus smallest among the DNA virus. So parvovirus is single stranded and smallest DNA virus. Hence, smallest DNA virus is parvovirus. All the DNA viruses replicate in nucleus as DNA is inside the nucleus. That's how on microscopy they are seen as intranuclear inclusions, except for pox virus. Think fox as a big animal and small nucleus can't accommodate it. That's why it is in cytoplasm. So this is the point how to remember. On hint is only DNA virus living in cytoplasm is pox virus. So here we got 2 points hint. First is the smallest DNA virus, which one is parvovirus and it is single stranded. And the second point is the DNA only DNA virus living in cytoplasm is pox virus. So other all DNA viruses are in intranuclear. Intranuclear replicate, but DNA virus living in cytoplasm is pox virus. Note that whenever a virus mess up with the nucleus, the nuclear material becomes susceptible to mutations. That's why DNA viruses can cause cancer. For example, hepatitis B virus. Hepatitis B virus, which is hepatine virus, uh, famous for hepatoma. Similarly, human papilloma virus, Epstein-Barr virus, HHV8 all causes tumor. So the hint is DNA virus can lead to cancers. Now the tip of exam is whenever in exam you see cervical dysplasia in the question, look for human papilloma virus infection in answer. Until and unless other diagnosis given is beyond any doubt, and cauliflower lesion on the perineal region are caused by human papilloma virus HPV. Okay, and now we are going to talk about RNA virus, which is single-stranded and replicate in cytoplasm. So they are divided into positive-stranded, icosahedral, negative-stranded, helical, and enveloped. So positive-stranded icosahedral, this group is further categorized into naked and enveloped. Positive stated are further divided into naked and enveloped. So naked positive stated are Carl Pico hair. Carl Pico hair. There is a pneumonic. कैलिसी वायरस पिकोरना वायरस हेयर में एच ई से बनेगा हेपी वायरस विच इज हेपेटाइटिस ई वायरस रियो वायरस आर ई रियो वायरस इज रोटा वायरस इन्वेलप्ड में टोगस CRF jars of colonic renal failure. So CRF. Toga virus, which is rubella and chikungunya. I sorry for that. I didn't pronounce it well. So CRF say coronavirus, SARS, retrovirus, HIV, flavivirus, HCV, dengue, and Zika. So reovirus is naked positive stranded, and retrovirus is enveloped positive stranded. Now we are talk about negative stranded, helical enveloped. Pneumonic is bad prof. Bad prof are negative stranded RNA virus. B B Bunya virus, which is Congo virus. Arena virus, Lassa fever, Delta virus, hepatitis D virus. For I just read it, don't need to panic. We just need to remember DNA virus. These are just for reading to have some short-term memory in them. So, so para-influenza, RSV, measles, mumps, and the prof R say rhabdovirus, rabies. O say orthomexovirus, influenza, and flavivirus, Ebola. So, okay, important points is exception to the rules are always there, but here is generalization to make the things easier and comfortable. So RNA wise is a big family stuff, but a single read is must. Don't get scared. Just a glance is enough. So here the author also said what I said to you. From above discussion, it is clear that whenever there is negatively stated, it is helical, enveloped, and RNA virus as a general rule. Okay? Memorize it by mnemonic bad prof or bad professor, while positive stated are icosahedral in structure and maybe either naked or enveloped, DNA or RNA. The coronavirus family has important viruses in its family, like three common among them are APC, hepatitis A, poliovirus, Coxsackie virus. These are APC virus. We say them APC. These are APC are coronavirus. Hepatitis A, polio, and Coxsackie virus. These are coronavirus. Per microscopy, RNA virus will show intracytoplasmic inclusion are most of as most of the RNA is in cytoplasm. These viruses will be single-stranded as RNA is single-stranded. Some exceptions are retrovirus will replicate in the nucleus, while reovirus will be double-stranded. So in RNA virus, as in DNA virus, we did the exception. Here in RNA virus also have exception. Like retrovirus is replicate in nucleus, not in cytoplasm. And reovirus is double-stranded, not single-stranded. So segmental RNA viruses can be memorized by mnemonic board. Virus or orthomyxovirus, arenavirus, and reovirus, which is reovirus is double-stranded. Stranded. Okay. So for board exam, Rio and orthomyxo at least should be remembered. Alhamdulillah. So, hint is segmental RNA virus, Rio and orthomyxovirus. Just read the RNA and important point. Okay, so this is minimum you should know about virology. Otherwise, it's a major field and books fall short for it. Concept of positive and negative strands. Positive means you have messenger RNA ready to produce proteins and to cause infection. That is, to get infection as you get the virus. While negative strand is vice versa, no messenger RNA at that time, and it will take up to 3 weeks to synthesize messenger RNA. Generally speaking, if a virus causes infection in less than a week, it's positive. So here we got a point. If it is causing disease in less than a week, so it's positive. Why positive? Because you have already messenger RNA. Mm, so it produces proteins and cause infection. When we get the virus, we get the infection. Its general concept and variation do occur between various viruses, but understanding it is important. Okay. Some highly tested viral disease. So just mug it up. Hand, foot, mouth disease. Hand, foot, mouth disease is caused by Coxsackie virus. Coxsackie virus, which is picorna virus, right? APC से आके. Hand, foot, mouth disease is caused by Coxsackie virus. Coxsackie virus type A. And hydrops fetalis is caused by parvovirus. Okay, so hand foot disease is caused by Coxsackie type A, and hydrops fetalis is caused by parvovirus. So how to solve hepatitis B viral profile related problem? Now the general rule. First of all, antigen will appear, that will be followed by antibody against it. So IgM shows acute or recent infection, while IgG indicates prior infection or chronic problem. That is, it's a antibody of chronicity. So what we just read, first of all, antigen will appear, then followed by antibody against it. And IgM antibody shows acute or recent infection, while IgG indicates prior infection or chronic problem. So the antibody of chronicity is IgG antibody. And now the important antigens antibodies of hepatitis B infection. HBC antigen. So first is HBC antigen. It is the first antigen to appear. HBC antigen is the first antigen to appear, but it is not usually detected because it appears during incubation period, and by the time of appearance of sign and symptoms it is gone. So what the point you have to learn? Never try to look for it in exams or clinical practice. Just understand it and ignore it. So just ignore it, okay? Because it's appear in incubation period antigen hepatitis, sorry, HBC antigen is appear during incubation period. So when sign and symptoms appear and the patient goes to doctor, then don't test for it. It will never appear in the test. So, because it's gone now. Anti HBC. So this is how we can look for it, that if antibody is present. So obviously antigen was present before. So anti HBC is the antibody against HBC antigen and appears 2 months after exposure. It can always be detected in the blood after exposure to hepatitis B. It is always be detected in the blood, okay? Anti-hbc is always be detected in the blood after exposure to hepatitis B. If anti-hbc is IgM, then the infection is acute. If IgG, it is chronic. Now that it is always positive after infection, even positive in window period. So the point is always look for it. If you are looking for previous exposure, it's negative only in immunization. It is negative only in immunization in our immunized individual, because immunization is not infection. So it is present in case of infection, not in case of immunization. So hint is anti-HBC IgM antibody acute, anti-HBC IgG chronic. So the next antigen is HBS antigen. HBs surface. So it is found on the surface of hepatitis B virus and indicates infection, whether acute or chronic. Decide chronicity on time duration, more than 6 months, or by looking at anti-HBC if it is IgM acute and IgG chronic. So HBs antigen is found on the surface of hepatitis B virus, and it is just indicate that infection is there, not tell whether it is acute or chronic. And then how we know about the acute or chronic? So look for anti HBC, which is antibody, is if anti HBC IgM, then acute, and IgG chronic, right? So HBS antigen is for infection. Anti-HBs, which is the antibody against HBs antigen, and it indicates immunity to hepatitis or recovery. So, as HBs antigen is for infection, so इसकी antibody किसकी आएगी? Indicates immunity. अगर anti-HBs positive है, it's mean there is immunity or recovery. Note that the time between the appearance of anti-HBs and disappearance of HBs antigen is called window period. So what is window period? Or serologic gap between disappearance of HBs antigen and appearance of anti-HBs. The next antigen is HBe antigen. It It appears after HBs antigen, and it is marker of acute viral replication. Marker of acute viral replications. When it is positive, there is high chance of infectivity. So HBs antigen is for infection, HBS antibody is for recovery and immunization. HBE antigen is for infectivity. If it is present, it means there is high chance of infectivity, and it is marker of acute viral replication. मतलब replicate हो रहा है. Hint HBE antigen E infectivity, marker of infectivity. Think it is an envelope that virus uses to send its infection. Hepatitis E, E is envelope. Think it is an envelope that virus uses to send its infection to the host. So whenever it is positive, stay away. The virus can send infection to you through this envelope. Anti-HBE. It appears after HBE antigen and it indicates low transmissibility of infection. So easy. HBE antigen is for high chance of infectivity. तो इसके अगेंस्ट एंटीबॉडी आई तो फिर इस इन्फेक्टिविटी को कम करना चाहिए ना. सो लो ट्रांसमिसिबिलिटी ऑफ infection. The order is HBC antigen, HBS antigen, HBE antigen. सबसे पहले C, then S and then E. HBC तो आपको नहीं मिलेगी because it in occur in antigen. HBC antigen is in incubation period. So इसके against जो antibody आएगी वो क्या है? Anti HBC. So it when if it shows whether um, acute or chronic infection. IgM, IgE, IgG से मालूम करेंगे. अब जो second antigen आएगा वो HBS antigen, which shows the patient have got infection and its antibodies against it is HBS, anti-HBS. So anti-HBS is when appear जब HBS antigen चला जाएगा and it shows immunization and recovery, and window period is between disappearance of HBS antigen and appearance of HBS antibody. The third and the last antibody we discussed, antigen and antibody discussed is HBE antigen. HBE in antigen shows infectivity. How you remembered it? E is like enveloped virus and enveloped to cause you infectivity for to cause infection or infectivity or trans transmission of disease. So if you have got HBE antigen in your blood, then it shows is marker of it's the marker of acute viral replication. It's replicated through its envelope, like how you remember E. So here it is high chance of infectivity. And anti-HBE, which is the antibody against HBE antigen, it shows low transmissibility of infection. So summary is HBS antigen, marker of infection, whether acute or chronic. HBE antigen is marker of transmissibility or infectivity. So run away here. It's how you remembered in exam because there are so many MCQs and we just remember in this way. So anti-HBE is marker of low or no infectivity. Anti-HBS marker of recovery from infection or immunization. Anti-HBC, which is the first antibody appear, always positive if the person is infected, if it is IgM acute, and if it is IgG chronic. As immunization is not infection, it will be negative in immunized individuals. So it's a point to I just missed it out while um, revising. So it's a point that anti-HBC is negative due in immunization, because immunization is not infection; it is on positive ish in infection. So important conditions. Some important conditions are: on the basis of above discussion, let's solve some important HBV profiles. Acute infection, C its infection, then HBs antigen should be positive, and acute, so anti-HBC IgM should be also be positive. And in acute cases, the infectivity is high, so HBe antigen should be positive as well. In chronic inflammation, sorry, in chronic infection and high infectivity, so C is in for infection we see hepatitis B surface antigen, HBs antigen should be positive, and as it is chronic, so anti-HBC IgM antibody should be positive, and high infectivity mean HBe antigen should be positive. Chronic infection, low infectivity, so here as infection is present, so HBs antigen positive, and it is chronic, so HBC antibody positive, IgG should be positive, and low infectivity, so anti-HBe should be anti-HBe for low transmissibility. अगर हाई इन्फेक्टिविटी है तो एंटीजन पॉजिटिव HBe एंटीजन, एंड लो इन्फेक्टिविटी है तो एंटीबॉडी HBe एंटीबॉडी पॉजिटिव. In case of recovery, so recovery is not infection, so HBs antigen should be negative. Antigen is negative, and it is recovery, so antibody HBs antibody is positive. There is no infectivity, so anti-HBe must be positive. It is not acute, so anti-HBC IgG should be positive. Note for further details, see table at the end. So just remember these points. It will help in exam, inshallah.